as we, inspired by Dr. Folty and her work, we continue to celebrate the theme of women leadership, women in cancer research, and women entrepreneurship in cancer. So with that, I want to introduce two, now I call friends, dear friends, Dr. Pam Garzone and Dr. Dimitri Giannakakou. I met Pam 5 years ago at the inaugural AIM High Salisbury, NFCR AIM High Salisbury competition. And that's where we had started to hatch the idea of a women's venture competition because of the underrepresentation of women entrepreneurs being noticed and being given funding. And she has been a catalyst, along with many, driving this program forward. And then Dr. Dimitra joined us. So the two of them have a lot of titles. I won't repeat. Chief Development Officer, I think. Next, general partner at Ventures. They do science, business, investment, entrepreneurship, the whole 9 yards. So I'll just give the stage to them.
Thank you so much, Rose. Appreciate the comments. And I'd like to thank Matt Tremblay, because without Matt giving up his space, and I would have not been able to participate in the Salisbury Award and meet all these fantastic individuals that are associated with NFCR and AIM High. So thanks, Matt. I appreciate it. So I am honored to serve as co-chair with my partner, Dr. Raju Kuchilapati, who's the other co-chair of the selection committee. The selection committee is comprised of esteemed researchers, clinicians, investors, and drug developers. And every year since it's the inception in 2020, the number of applications has risen. And every year, I think we're at our peak of seeing, the innovativeness, the quality of the applications, or the solutions to our high unmet medical needs for cancer patients. And every year, it just keeps getting better and better. The creativity and the passion of the entrepreneurs that apply to this award is just amazing. And this year, we had 84 applicants, representing mostly therapeutics, but also diagnostics, digital health, and delivery of drugs, as well as some research tools. And we had applicants from 14 different countries, including the US, and they represented India, and Singapore, several of the Asian Pacific regions, Israel, Europe, and Africa. And so, again, to speak to the outreach of NFCR and AIM High, it's really global. The process, how we select, we have our selection committee reviews all the applicants. 2 committee members are assigned to each application, and there's scores given, and we meet as a group and discuss those scores, discuss why we scored one way or another. And from that, all those applicants, we narrow the focus down to 8 to 10 semifinalists. Those 8 to 10 semifinalists are given the opportunity to pitch to the entire selection committee. Again, additional scoring is done, and from that group, there are winners that are selected. And this year, I am so happy to present the AIM High Award to Nabanita Nawar. She's the co-founder and CEO of HDAX Therapeutics, whose innovative 2-site binding technology delivers high binding affinity and long duration of action. They're initially focusing on HDAX6 inhibitors. And it's just, it was a great presentation, both in the deck as well as in the video, and we are pleased to award her the Aim High Women's Venture Competition. Anita?
Congratulations.
On behalf of Aim High.
Yeah.
Now I will turn it over to Dimitra to give the award for the other winner of the competition.
Thank you so much, Pam. And I think it's my turn to thank Pam Garzone, because of her, I was invited to be part of the Women's Venture Competition. So you see how we always have to give back and lift how we rise. It's been an honor to be in the last few years part of this organization. Dr. Sujwanpa, Rose have been amazing partners. I am the head of the investment committee from the Women's Venture Competition, and I am chaired together with Dr. Jimmy Liu, my partner, so he's there in the audience. And we had the hard job to actually review the top winners for considerations for an investment. We also have a process. We have an investment memo that we have to write. We have factors that we go beyond the research and the science that our selection committee had to have the top winners. And with that, we go through the process where we decide and make a recommendation to the Aim High leadership and FCR leadership for investments one way or another. But in the meantime, I want to tell you how we also wanted to consider the second winner, which is Professor Paraskevi Giannakakou. They invited me here because I can say her Greek name. So she is a professor at the Weill Cornell Medicine, and in her lab, she focuses on the development of anti-tumor drugs that target microtubules. She got her Ph.D. in the Athens University Medical School, and I said I will say that, go Greece. And then she also did her postdoc with NCI. But she's also the CEO of ARMA BIO, who's the second winner of our competition. And in that company, they have developed a first-in-class molecular glue that is actually developed for advanced prostate cancer patients that develop drug resistance. So it's very exciting to see what they're going to do next. And with that, I would like to invite Dr. Paraskevi Giannakakou up to the stage.
Sherry is also co-founder of the company. Sherry, come on. We're a women-led company, so just want to emphasize that. And Sherry is co-founder and assistant professor at Harvard Medical School. Thank you.
Let's give these two women a great round of applause.
Hi, everyone. I'm Nabanita. I'm the CEO and co-founder of HDAX Therapeutics. Firstly, thank you so much for this wonderful opportunity and recognition. We're really humbled and honored to win the first prize with the distinction of Aim High. Today, I'll tell you a little bit about what we're building at HDAX. At HDAX, we are developing next-generation small molecule therapeutics targeting HDAX6 to really transform patient outcomes. Over the years, researchers have learned about the molecular mechanisms behind transport problems in the body. And one disease target that has stood out is this protein called HDAX6. HDAX6 is a key protein involved in multiple pathways in the body. It deacylates alpha-tubulin, making it really important in microtubule and axon transport, and also cytoskeleton structure, and also mitochondrial regulation and inflammation, making it implicated in a wide range of disease areas, starting from peripheral neuropathies that happen in cancer patients from chemotherapy to cardiometabolic diseases such as heart failure with preserved ejection fraction. This has been an exciting and somewhat validated therapeutic target for a while. However, the field has suffered from a lot of challenges because we're still using the same decade-old approach in going after this really important disease driver protein. And the three main challenges we see with current technology starts with challenges with efficacy, which arises because of poor target engagement at your molecular level with HDAX6. Next, challenges of safety. HDAX6 falls in a family of 11 proteins. There's HDAC1, 3, 10. So being able to target HDAX6 selectively and specifically is actually not a trivial task at all. And existing molecules, FDA-approved drugs, and even molecules that are in development today end up targeting multiple proteins at the same time, leading to adverse side effects in patients. Finally, challenges with pharmacokinetics. Current molecules have a very hard time surviving in the body and also being able to reach the site of action for the right duration. For chemo-induced peripheral neuropathy in cancer patients, a lot of these patients also suffer from cognitive impairment. So blood-brain barrier permeability is also required to some extent in order to really address this patient population, and that has been very difficult to get. So at the University of Toronto over the last 7-8 years, and now at HDAX Therapeutics for the last 3 years, we've been building a solution not just for one, but in fact all three of these challenges. What we have developed over the years is a novel and differentiated two-site binding mechanism to target HDAX6. So what I have here is an X-ray crystal structure of one of our tool compounds in the pocket of the protein HDAX6. This is proof of principle paper was published in JMedChem. This first arrow shows the first interaction. This is how typically all current molecules engage in the target. It binds to the zinc in this metalloprotein. At HDAX, we've built on a second layer of interaction. So simply put, we can grasp onto the target with both hands, while others can only do so with just one hand. This bitopic binding mechanism really allows us to engage the target much stronger and much longer. And that has led to a lot of competitive advantages in contrast to what's currently out there for patients and also what's being developed. In contrast to the first generation HDAX6 inhibitors, we've been able to show much stronger target engagement, much lower IC50s for HDAX6, but also very importantly, binding kinetics. We actually engage the target much longer than singular binders. Then that really helps us improve efficacy, which is what we've shown in our animal studies. Next, being selective and safe and really having a good therapeutic window. We've been able to show almost 800-fold selectivity for the target, while the first generation of molecules were really poorly selective in cellular studies, and so even less selective in complex animal models and humans. And finally, we've got two programs, one that crosses the blood-brain barrier for cognitive impairment associated with chemo-induced peripheral neuropathy, and other neurological indications that are driven by microtubule dysfunction. And we also have a peripherally selective program for peripheral indications. And that's what I was mentioning. The current pipeline explores use of our distinct molecules in two separate programs, central acting for chemo-induced peripheral neuropathy and also other other neuropathies and neurodegenerative diseases. And the second program is in the cardiometabolic space, both at the lead optimization stage right now. And our first shot on goal is chemo-induced peripheral neuropathy, a very underappreciated but a huge, huge unmet need, which I'm sure this whole room appreciates. 30 to 80% of all cancer patients, regardless of what cancer, regardless of what treatment we're giving or which chemotherapy, ends up developing some level of peripheral neuropathy. It starts with pain, numbness, burning sensation in your fingers and toes, but for some people, it can be extremely debilitating pain, immobility, and also partial or full paralysis. For some cancer patients, this can actually be permanent if the nerve damage is too severe. And this is caused by chemotherapy, but also immune checkpoint inhibitors, antibody drug conjugates, and there's really no cure for this. Currently, the standard of care is just symptom management. So put your fingers and toes in a bucket of ice, which really has nothing to do with the biological problem, the nerve damage, the transport dysfunction that's happening because of the toxic chemotherapy. And we now understand how this happens, how HDAX6, given its role in axon transport, de-acetylation of alpha tubulin in your microtubule, how its inhibition can actually reverse this nerve damage, but we are yet to see strong drug candidates that can truly reverse this disease. So at HDAX, we are pioneering the first disease-modifying therapy for chemotherapy-induced peripheral neuropathy in cancer patients. We have some very exciting data on reversal of pain, numbness, executive functioning, and also reversal of memory decline. This is our team. We're scientists, we are entrepreneurs, we're clinicians, and serial entrepreneurs and seasoned board members all coming together to really take these therapies towards patients. We have actually recently closed our seed financing, and very happy to say that AIM High has participated and Pamela has joined as a board observer. In short, at HDAX, we are developing next generation therapeutics for diseases out of a microtubule dysfunction, starting with chemotherapy-induced peripheral neuropathy in cancer patients. Thank you for your time.
So thank you for the opportunity to present here today. When I was invited to this meeting, I did not know what to expect, and I'm really wowed by the scientific and intellectual caliber in this conference. Fantastic. So I'm going to talk to you about our first-in-class molecular glue degrader for prostate cancer therapeutics. So why prostate cancer? So this is our company's co-founders. So prostate cancer, as you all know, is the most commonly diagnosed and second leading cause of cancer death in men in the US. And it is actually more common than you think. 1 in 8 men will have prostate cancer at some time during their lifetime, while the recent Lancet Commission on Prostate Cancer report published in August 2024 projects that newly diagnosed cases of prostate cancer will rise from 1.4 million in 2020 to 2.9 million men in 2040. So it is a huge problem. So prostate cancer is driven by androgen receptor signaling, and currently 90% of the drugs in prostate cancer target AR. So treatment resistance, however, invariably occurs and leaves patients with limited therapeutic options. And one variant of the androgen receptor, AR variant 7, is a leading cause of treatment resistance and is currently undruggable. So we performed a high-throughput phenotypic screen of 170,000 compounds, identified our compound, which is first-in-class molecular glue degrader for both ARV7 and AR. And we think the impact of our drug is it has the potential to replace existing AR inhibitor therapies by inhibiting AR, which is 90% of current therapies, and also delay the onset of resistance by also taking out ARV7. With that, I was asked to put this slide in. So currently, we're very early stage. We have gotten $3 or $4 million through different funds, especially NCI, STTR, SBIR. So we're seeking currently $15 million to accelerate to drug candidate determination and IND filing. Okay, so as I told you before, on the left, you see the market sales for prostate cancer therapeutics. AR-directed therapies claim 90% of the current therapeutic space in prostate cancer, and it is projected to claim the majority by 2030. So AR-directed therapies target the ligand binding domain of AR. A direct result of this targeting is that you get expressions of AR splice variants, which lack now the ligand binding domain and confer resistance to all standard of care treatment. So the clinical challenge and the unmet need is that ARV7 is constitutively active and confers clinical drug resistance. So we focused on ARV7 among the other AR variants is the most prevalent, expressed conservatively in 75% of patients with metastatic castration-resistant prostate cancer. It is constitutively active in the nucleus, and when you look at the functional domains, you realize that V7 lacks the ligand binding domain and the hinge domain, which we showed mediates taxane sensitivity. Taxane is the only chemotherapy that works in prostate cancer, and ARV7 confers resistance to both. In addition, in 2020, the FDA approved olaparib PARP inhibitors. ARV7 was shown to activate DNA damage response pathway, also confer resistance to PARP inhibitors. And so it is currently undruggable, and to address this need, we performed a physical phenotypic screen of 170,000 small molecules using ARV7 nuclear localization as the assay endpoint. We identified our hit molecules that inhibit ARV7, and early on, we knew that we have a unique chemotype as a starting point, completely different than all current AR inhibitors in the space, preclinical and clinical space, compared to conventional, non-conventional AR inhibitors, and so on. So then, mechanistically, our hit compound destabilized ARV7 by shortening its half-life, and we found that this mechanism involved ubiquitin proteasome pathway. So we knew we were dealing with a degrader. We engaged in medicinal chemistry. We had 60 analogs synthesized around the functional domains. We tested them in a single dose titration, identified the candidate drugs by EC50, and based on the EC50 results, drug likeness scores, and our in vivo PK results, we focused on two lead compounds, 15 and 64, for further development. So for the chemists in the group, you know that the moment you change even a single atom in a molecule, it may have a completely different mechanism of action. So to make sure we're dealing with the same mechanism, we saw that compound 15 inhibits ARV7 within the first 4 hours, so very quickly, through the proteasome, and we know that the family of Coulin ring E3 ligase are required for the activity while it does not affect AR transcription. Furthermore, in preclinical models, we show here that compound 15 reverses enzalutamide resistance in cells that express both AR and ARV7, but has no activity in normal prostate epithelial cells. It does not have activity in AR null prostate cancer cells, and so on. Also, both compounds are much more active in the same cell line, where enzalutamide and the Arvinas, ARV1110, AR full-length product shows no activity. And in cells that have only AR, no ARV7, our compounds are more active than both enzalutamide and ARV. So how does it work? We have preliminary cryo-EM data that show that our compound binds directly AR and changes its conformation. And then to dissect the functional domains to see where it binds, we saw that the N-terminal domain of AR, which is highly disordered, mediates the activity of our compound. And not only that, within the N-terminal domain, AF1 and Tau1 are the sites that mediate the activity and no Tau5. Currently, there is no drug that binds Tau1, and there is no N-terminal binder that degrades AR. So this is another unique aspect of ARs. So we know that it is intrinsically disordered, but we engaged in some modeling using Rosetta Fold 2. AlphaFold did not work for this disorder domain, and Rosetta DNA that you add the DNA interactions to stabilize the proteins. To validate the assay internally, we just modeled enzalutamide, which shown to bind as expected in the ligand binding domain and be in direct contact with phenylalanine 876. It is a known clinical mutation that confers resistance to enzalutamide and an analog apalutamide. So doing the same for the AR N-terminus, we show that our compound binds at the N-terminus in the domain that overlaps between Tau1 and AF1, consistent with our preliminary data. We're obviously improving on this analysis while we're seeking to have single molecule cryo-EM determination, protein NMR, and so on. So compared to the current landscape, our compound is the only one that targets both AR full length and ARV7. And we're trying to find the E3 ligase that mediates the activity by CRISPR E3 ligase screening, proximity ligation TurboID screening, etc. And right now, we are, I do not know if I have a pointer, but we are in the middle point where it says 2025, where we're bringing this for in vivo efficacy data. And that's it. Thank you so much. Okay, done.